One, two, three, four, five. Counting seems easy, right? Well, try doing that 10,000 times without messing up. See, important to the work of biology is cell counting. While in small batches it seems easy, at the scale for research it becomes quite daunting. Thus I, Tyler Jane, and my fellow authors present a new tool to make the jobs of researchers a little bit easier. So, why count cells? For many studies, we want to know how well our medical treatment worked. For an example, in my lab, we focus on treating HIV. One way to know how successful a treatment was is by looking at the number of surviving cells in treated versus untreated cell tissue. This process can give you very useful statistical information on the success of the treatment. However, this is extremely tedious if you're hand counting hundreds or thousands of cells. So rather than asking an undergraduate student to arduously count cells for a month, how about using a computer? So while machine learning based methods to automate this process do exist, they have several issues. One is that they're often inaccessible due to prerequisite technical knowledge. While this audience might be familiar with command line terminals and running scripts, biologists often aren't. Additionally, such tools require high computational power. Not everyone has access to large computer clusters, which are large networks of computers that allow you to have high computational power. Thus, we introduce ACCT, a user-friendly tool for automatic cell counting through image segmentation following user-driven training. So ACCT expands on the current open-source Trainable Weka segmentation package, available on the Fiji distribution of ImageJ. ImageJ is a tool used often in biological research to look at images, especially of cells, for information. The entire ACCT study was conducted using consumer-grade laptops, such as the Dell Inspiron, to demonstrate that low computational resources are necessary to perform our automatic tool. So we evaluate ACCT against two different published image data sets, comparing the automatic counts of ACCT versus manual counts from human observers. First is the IVA1 dataset, which has images of mouse cortex tissue containing microglia stained for IVA1, which is a microglial marker. Next, we have FluoCell, which contains images of mice brain tissue, which are stained with fluorescent dye, which allows us to visualize neurons inside of images. So we separate these images into three different subsets: training, validation, and the unseen full image set. The training set is used to generate classifiers, which are the machine learning models that will eventually count the cells inside of each image. We check how well these models we created perform against the validation data set, which allows us to determine the precision, accuracy, and other metrics to see how well these classifiers perform. Finally, we have the unseen full image set, which is the full set of images we want to count for cells. So ACCT implements several different machine learning models. These models essentially will classify pixels in an image as belonging to cell or non-cell. We, in this study, look at these two models, fast random forest and BaseNet. However, ACCT contains many more models users can choose from, and users are able to upload their own models to ACCT as long as they're compatible with ImageJ. So we are able to then select features to train the model. Some are listed on the right. These features tells the model what exactly it's looking for when it's being trained. For example, one such value is maximum, which means that the model will look at the maximum intensity value or brightness of selected pixels when training. Thus, if the pixels of your cells in the image are really bright, selecting for a maximum intensity would help with training. So here's an example of the GUI interface that users interact with when training classifiers for cell counting. On the left, we see that cell pixels are marked in red, and the background is marked in green. in this darker red were the exact cell pixels a user selected when trying to train a classifier. And in the lighter greens highlight, we see the pixels the user selected to the marked background. Classifier then trains the rest of the image, filling in the rest of the red and green for where it believes there are cell and background pixels. We can also see this represented on the right, where yellow dots represent the center of a cell, and for ease of clarity in this demonstration, we see circled with this big red circle. So doing this repeatedly back and forth creates more and more classifiers in iterative steps. This allows the users to make adjustments and corrections to make their model a bit more accurate every single time. Doing so allows us to have more accurate classifiers for precision and accuracy, which the user will want to care about when selecting the best classifier when counting their full set of images. So this figure depicts an image taken from the FluoCell dataset before and after segmentation. The cells in the original image are indicated by the bright yellow fluorescent colors, and the resulting segmentation depicts where a certain classifier thought cell pixels are located and thus where cells are. The overall workflow of ACT is represented here. Training images are used to generate classifiers, which are then measured against the validation images. From here, we looked at the statistical performance on each of these classifiers to choose the best classifier. This is left to the user, but it's likely the classifier with the greatest precision, greatest accuracy, or in our case, greatest F1 score, which is harmonic mean for precision and recall. This gives us a best classifier used to count the unseen images, which generate segmented counted images, and statistical information is reported on both the number of cells and morphological information of the cells. So this is an example of results from the validation stage. This is classified performance on the IVA1 validation images, and we sort them by F1 scores. This shows how the best classifier can be determined. Here we see classifier 4, 2, and 10 have the highest F1 scores, while classifier 17, 1, and 3 have the lowest. We see here that we also report the mean average error and mean percent error, which are the raw number deviations from the manual counts versus the automatic counts. Next, we demonstrate correlation analysis of microglial density. The idea here is we want to show how human-like is ACT when counted, compared to two human observers, observer A and observer B. So we see the number of cells counted in two different experimental conditions, resting and activated, are similar with a high adjusted R value between classifier 10 and observer A and observer B. So much so that the correlation between our ACT classifier was greater than the two humans between each other. Thus, ACT is able to show a human-like differentiation in cell counting between experimental conditions, thus showing it's able to represent experiments. Here, example of the performance of two ACT classifiers on IVA1 images, classifier 4 and classifier 10, which are both fast random forest models. We see here that in the IVA1 dataset, we have high levels of precision and recall, tends to be higher in precision. This is shown on the validation, full dataset, and audit dataset. The audit dataset is a subset of the full dataset which is used for statistical analysis in our study. Next, we have the performance of ACT classifiers on the FluoCell images. Here is a fast random forest model and a Bayes network model. You see here slightly lower performance compared to the IVA1 dataset. Overall, we're able to count cells similar to trained researchers using ACT. And when we compare the performance of recall scores, we tend to do better on precision. And when comparing the two data sets, we see that IVA1 images had more consistent cell density, intensity, and fluorescence. So ACT is likely to be better performed on that data set. Overall, ACT uses significantly less time than manually counting cells, requires low computational power, and because it's using a GUI, it makes it easy to learn for those with low computational background. Thus, with ACT, an open source tool, this should help researchers in the field overall.